submit your questions anytime throughout the talk. And any questions that are not answered during this session will be answered by the presenter in a follow-up email. Closed captioning for this meeting is available and for your use, and this session is being recorded as well. So lastly, if you're having technical difficulty, please email headquarters@aro.org for assistance. And now, without further delay, I would like to introduce you, Dr. Richardson. So Dr. Richardson, welcome, and it's a pleasure to have you here.
Thank you, Melissa. It's really exciting to be here with all of you today, virtually, of course. It would be obviously great to be with you in person, but this is definitely the next best thing. I'd really love to talk to you about some of the work that we're doing at the Bionics Institute. And one of these projects is all about the new technology that we could use for improving the precision of cochlear implants. So at the Bionics Institute, we're interested in neuromodulation via bionic devices for a whole host of indications. Pretty much if there's a nerve in the body that's accessible for an implant, and if it can be used to treat neurological disease, then we're interested in doing research in that area. But of course, for this crowd, for this audience, it's the cochlear implant and hearing loss that's the most relevant. So that's what I'm going to talk about. And as you all probably very well know, in the normal auditory system, the basilar membrane and the organ of Corti act as a mechanical spectrum analyzer, such that the high frequency information is represented primarily in the base of the cochlea, and the low frequency information is represented mainly in the apex. The signal is composed of multiple frequencies, and the resulting traveling wave will create maximum displacement at different points along the basilar membrane. The spectral information is also encoded in the temporal firing properties of auditory neurons. So nerve fibers fire at rates that are proportional to the period of the input signal of up to frequencies of 5 kilohertz. And at low frequencies, individual nerve fibers fire at each cycle of the stimulus. So that is they're phase locked with the stimulus. And at the higher frequencies, this is indicated by the organized firing of groups of nerve fibers. So in cochlear implant recipients with severe to profound hearing loss, the function of the basilar membrane and sensory cells is lost, and consequently the implant stimulation pattern must encode and deliver the spectral and temporal information to the surviving auditory neurons. So the incoming spectrum is divided and mapped to the individual electrodes, and once again, the high frequency information is mapped to the more basal electrodes, and the low frequency is mapped to the more apical electrodes. And the temporal information is represented by modulating the stimulating pulse train with the envelope of the signal within each frequency band. So cochlear implant recipients can use the temporal and place mechanisms for pitch discriminations, and in fact, subjects tend to report higher pitches as the stimulating frequency is increased and as the stimulating electrode is moved towards the base. But the number of pitches of the cochlear implant is limited by the number and location of the stimulating channels, and current spread from each electrode does result in a low spatial resolution. So if we have 2 neighboring electrodes and if they are both stimulated at once, a nerve that lies, let's say, in this place, can receive the stimulating current from any one of those electrodes. And this means that they're not independent channels, and that can lead to sort of uncontrolled loudness. And it's very difficult to deliver the fine temporal structure of the stimulus. And so with channel interactions, this is reducing the number of independent channels in the implant to about 8. So one of the questions that we've been asking is, well, can we increase the number of independent stimulating channels? So to look into technologies that might improve the issue of current spread, we need to be able to measure spread of activation, just because all of the research I'm talking about is in preclinical animal models, such as the mouse or guinea pig. So we can record from the brain to see the current spread in the cochlea, and we use the inferior colliculus because it's quite accessible. In the mouse, it's just sitting there right on the top. And the tonotopic map of the cochlea is preserved throughout the auditory pathway. So if we're looking at the base of the cochlea here, which is represented in purple, and the apex, which is in red, that tonotopic map is conserved such that in the inferior colliculus, the apex or the red area of the cochlea is on these more superficial recording sites, and the base of the cochlea or the purple area is represented in these deeper recording sites. So if we have then a multi-channel electrode that's positioned in the inferior colliculus, we'll be able to see like a map of the area of the cochlea that's being activated. And so the way this happens, the way we can, or what we see from these recording electrodes is that we record multi-unit responses. And this is a trace of the multi-unit responses where you see the stimulation artifact. And by the way, a lot of these images are borrowed from Chef and George, who did some really nice work in this area. So thanks for that, Chef. And so I was saying, you can see the stimulation artifact from the electrical stimulation and the multi-unit responses in this analysis window, which is, you can set that, say, from 3 to 35 milliseconds. And we can generate a post-stimulus time histogram of the spikes during stimulation of the implant, showing the early onset responses of the neurons in the inferior colliculus. And if we look at the normalized spike rate versus the stimulus intensity function of the best recording site is, say, the one with the lowest threshold. This is where we can take measurements such as the threshold, and also levels of discrimination such as threshold plus d prime equals 1, so that we can find equivalent regions above threshold to compare the spread of activation. So just because I will be using response images throughout the talk, so I just wanted to again highlight some of the key features that it's showing. So it is this normalized spiking activity where the maximum spike rate is given a yellow color and spontaneous activity is given 0 or the black color. And we can define threshold. Here we've defined it as normalized spike activity of 0.3. And this generates a spatial tuning curve across all the electrodes. And if you recall, the more superficial recording sites correspond to the apex of the cochlea, and those deeper recording sites correspond to the base of the cochlea. And so this gives us a little bit of a map of the areas of activation in the cochlea. And again, the width of the spatial tuning curve can be measured at equivalent areas, and I'll be using that d prime equals 1 or d prime equals 2, just as 2 levels of discrimination. And the best recording site again was that one with the lowest threshold. And so continuing to use Chef and George's examples of spatial resolution. Here we've got an acoustic stimulus and electrical stimulation. And of course, the acoustic stimulation gives this nice sharp spatial tuning curve, and the electrical stimulation had a very broad spatial tuning curve. This is given by monopolar stimulation. You see it's got the best recording site, but as soon as you turn up the current, you don't have to turn it up very much before you're getting really broad activation across just about all of the cochlear, and you turn it up a little bit more and you start to get myogenic activity. So it doesn't have a lot of range. A main aim of our research is to increase the independent stimulating channels, perhaps an order of magnitude. And again, I'll be focusing on that preclinical research methods rather than the clinical studies, such as current focusing methods for the electrical stimulation. But my main focus of the talk is these alternative methods of stimulation. Definitely want to acknowledge that you can use methods to focus the electrical stimulus, such as using focused multipolar stimulation, whereas monopolar stimulation, you're just applying current to that one electrode there with a remote return. With tripolar stimulation, you've got a positive current on one electrode and that negative current on the flanking electrodes. And this, as well as the focused multipolar stimulation, can really result in this very sharp spatial tuning curve. But when this is applied to clinical studies, there's an absence of any clear advantages of the tripolar configurations in contrast to what we see in the animal studies. And the differences in the speech performance were really just rather small and doesn't really justify the clinical introduction, and it's not very well taken up in the clinic. The perception of pitch can also be varied dramatically by adjusting the proportion of current delivered simultaneously to two electrodes. So in this one, for example, there's equal current going to most the two electrodes, and so the peak current is lying right in the middle. But if we apply a little bit more current to electrode 6 and a little bit less to 5, that current can be steered across and to create virtual electrodes. So if this is the original signal and these down the bottom are the electrodes that are in position, then with fixed channels, then this is the kind of pattern that can be used to try to match that signal. But if we can use virtual channels, then maybe we can more faithfully follow the original signal. And in fact, when this was done, and this is looking at the center of activation, I think it was in guinea pigs in this study, you can see that when the current is altered from 0 to 100, then that center of activity can be measurably moved across on that image there. But a big focus of my talk really is on these alternative technologies to electrical stimulation, where we can sort of really circumvent the issue of current spread. One of these is to use light. And the idea is that you've got a multi-channel implant that now consists of light emitters, and because it's not subject to current spread, that we might be able to achieve high spatial resolution. If we're using infrared light, there's no need to genetically modify the tissue, so that's a real advantage of that technique. It does use quite high powers, and the mechanism of action is that the infrared pulses are absorbed by water, causing a rapid but transient local heating. And that temperature increase induces a capacitive current, which depolarizes the cell membrane and induces action potentials. But heating can also generate a photoacoustic effect, which can also activate residual hair cells if they are there. So it's probably a combination of these mechanisms. But certainly in guinea pigs, when there's a laser-coupled optical fiber directed towards the auditory nerves, the spatial resolution is really quite nice. So this is that spatial tuning curve just turned on its side here. And you can see that the activity is recorded across just a very tight area of the recording electrodes there. And optogenetics is another technique to control the activity of neurons with light, but it does require the expression of light-sensitive ion channels in the target cells. And this can introduce selectivity. So whereas electrical stimulation, the current spread the electrodes and activates any of the nerves pathway. With optogenetic excitation, when you put the light beam through the tissue, only the nerves that are expressing the opsons will respond to the light. So that's that idea of getting selectivity from optogenetics. And but the light can be focused. I'd argue perhaps a little bit more easily than electrical current can be focused. So that can introduce some precision. And it does use lower power compared to infrared radiation. And there's been some really nice pioneering work from Tobias Moser's lab in Göttingen, where the opsons have been expressed in the spiral ganglion neurons. And they've achieved really, really nice solution there. So compared to monopolar stimulation, which is activating very broadly, the cochlear or colliculus, it's really quite nice with the optical stimulation and quite similar to the acoustic stimulus. Opsons come in a whole range of properties. There's dozens of them. They can have different activation wavelengths. So I've been always showing this example with blue light, which is coming in and changing the conformation of the ion channel there and letting ions flow into the cell. But it doesn't have to be blue light, and there are some that are certainly more red shifted, such as crimson. And they also come with a range of opening and closing kinetics, and this is showing the closing kinetics. And you can see that some are very fast, like Kronos, and some of the more, I would call, moderate rate, such as the channel rhodopsin 2, and some that are very, very slow, minutes to close, and that's the step function opsons over here. So when we're using optogenetics, we need some animal models. I'm throughout this talk using mice, but it doesn't have to be mice. But for example, there are transgenic mice that you can make to express the opson of your choice. And throughout this talk, I'm going to use this one here, which is it's channel rhodopsin 2 with the H134R variant, which is one of the slower opsons. It's actually even a little bit slower than channel rhodopsin 2. And in the transgenic mouse, of course, you can use a promoter to express the opson wherever you like. Synapsin promoters are quite good for spiral ganglion neurons. But my mouse model is using parvalbumin, which means it is expressed in the spiral ganglion neurons, but also in the hair cells. So we do have to be careful to ensure that we wipe out all the hair cells before we do any of our studies. It has a peak activation with blue light and these moderately slow closing kinetics of 18 milliseconds. And also transgenic mouse is not very translatable. So we've also got the adeno-associated virus models where you inject the virus into the cochlea, again, using the promoter and opson of your choice. But just to note that the expression will be more variable than your transgenic mouse, so interpretation becomes a little bit more tricky. But using those mouse models, I just want to discuss some of the things around optogenetic stimulation, such as spectral resolution, temporal resolution. A really key thing I'd like to talk about is hybrid stimulation. We've been doing a lot of work around this in our labs. Very, very briefly, just development of translatable opson expression systems and the optical cochlear implants. Starting with the spectral resolution. The demonstration that we do get that cochlear topic activation with light. So we can place a laser coupled optical fiber either into the basal turn, just by positioning it through the round window membrane, or by making a little hole in the apical turn and placing it into the apex of the cochlea. And you can get this shift in the best recording site from that basal area for the basal site over to the apical area for the apical fiber. So we are getting this cochleotopic activation, but just noting that for the basal turn, the light direction is not particularly optimized. So we do get this a little bit more spread because it's kind of being directed down the scala tympani rather than really being positioned towards the auditory nerves fibers. So to improve that in the basal turn, so comparing this laser coupled optical fiber now to an LED, which where the light is now coming out the side, we can definitely improve the width of the spatial tuning curve when we're using the LEDs. And that's just that LED compared to a single channel of electrical stimulation in the monopolar configuration here. So definitely, yes, we can improve the spatial resolution using light. So we were really interested in the channel interactions. And so we needed multi-channel devices. So we've got, for the mouse, an implant that has 4 electrodes, with electrode 1 being defined as the one that's going to be in the base of the cochlea, and the tip electrode, which is electrode 4. As well as a multi-channel LED device, which I've shown here before. It's been coated in silicone just so you can see it a little bit better. Again, with this one has 5 light emitters or micro LEDs. They're about 270 by 250 microns, and they've got, again, numbered LED 1 to LED 5. And this is the spatial tuning curves of those individual electrodes on the top row here, and the individual LEDs on the bottom row here. So just noting again, with the left side being more apical and the right side being more basal, you can see that the best electrode is shifting across the recording electrode array, both for the light and for the electrical, but that typically the LEDs are giving a narrower spatial tuning curve. And so we measured it at those 2 levels of discrimination above threshold, D prime equals 1 and D prime equals 2. And so for that first one, D prime equals 1, you can see that the optical stimulation is the spread of activation is less than half that of the polar stimulation. And we've just put acoustic here in normal hearing animals just for comparison, but these are different deafened animals for the electrical and optical stimulation. And also for D prime 2 above threshold, you can see the same thing happening there. And so this was some really nice work from a PhD student in the lab, Ajmal Aziz, as is the following work, where we're now looking at the channel interactions. So we're going to use the simultaneous stimulation with electrical and optical stimulation. And so first of all, we'll look at, I'll call it, say, channel 1, so electrode 1 for electrical, and LED 1 for the optical. And then we have a look at the next channel, channel 2, on its own, and we note the threshold, which is 160 current levels for electrical, or 2 milliamps for the optical. Then we stimulate them simultaneously, and for channel 1, we'll do that at a set intensity level, such as at threshold. And for channel 2, we'll we'll stimulate it at a range of frequencies and measure the influence of having electrode 1 activated. So for electrical stimulation, you can see the influence on threshold of having that channel 1 there at the same time. The threshold has dropped down to 100 current levels. So there is interaction between the channels. The light, when we stimulated them simultaneously, there was no influence of having that first light or LED 1 on the threshold for LED 2. We can use that at a range of set levels. So now I've got electrode 1 at a set level of threshold plus 20 dB together with E2, and you can see again a very big influence for the electrical stimulation, but for the optical stimulation it was still unaffected. So we can plot this threshold shift difference at those different set intensity levels. So we can use below threshold or at threshold or above threshold for that first electrode. And looking at the threshold shift, you can see for the below threshold, there's very little impact on the optical stimulation, but for electrical stimulation, particularly at these lower inter-electrode spacings, you can see that there's quite a big effect on the threshold. The same story for when we're using at threshold stimulation. There's very little influence on the optical stimulation except at these very close light spacings, but a huge influence on the electrical, and the same thing for the above threshold stimulation. So this is sort of our way of measuring that, those interactions between channels and being able to measure whether we can increase the number of independent stimulating channels. And so next we wanted to look at the temporal resolution of optogenetic stimulation. And so some of these studies were done in vitro, in patch clamped auditory neuron from our mouse. We do know that higher stimulation rates are needed to preserve the temporal information of sound that encodes pitch and sound quality. And so you can see in this recording, which was done at just 20 hertz and a presentation of 10 pulses of optical only stimulation, there's an action potential to the first pulse, and then there was no responses. And it's not too surprising. This, again, we're using this quite slow opsin here. It's not Chronos or anything. But looking in vivo in the mouse as well, what happens, we're recording these compound action potentials, 100 presented at 100 pulses per second. There's 30 presentations here, and you can see that the amplitude of the response declines throughout that burst. And if we overlay the response of the first and the last and some of the other ones in between, and you can see that there is the first here is plotted in blue and the last one is in purple. And you can see that there is a large decline in the amplitude of that response, as well as in the spread, spread. Whereas with electrical stimulation, it's pretty consistent throughout, and you can see that when we overlay the. So to measure that, this graph is showing the ratio of the last and the first response amplitude. So for electrical stimulation, it's 1, that is the amplitude change over the burst. And for optical stimulation, it's around 0.5. So there's that big effect on the amplitude. And this graph is showing the temporal spread. So for electrical stimulation, you recall that it's very low because when you overlaid it, it had barely changed. But for optical stimulation, it had worsened over the burst. And this graph is also showing the percentage of responses elicited at the 100 pulses per second. You can see that also declines. And again, so a lot of this is related to the use of the relatively slow opsin. Certainly some groups have been using Chronos, which is much faster, and we can certainly make some improvements. But we've also been thinking about other ways that we can improve the temporal response and stimulation. The main concept is that sorry, I'm just going to go back because I just wanted to highlight that this work was this is the work of Elise AJ, who's a PhD student in the lab as well. And she's going to be presenting some of these results at ARO for those of you who are attending. So if you want to know a little bit more about this, certainly see if you can go to Elise's talk. Sorry, moving on to hybrid stimulation. So the main concept is that the optical stimulation is used to raise the membrane potential and make the neuron more ready to fire. And then we can come in with a really small electrical stimulus to induce the action potentials. And so by using electrical to activate the neurons, we're aiming to get higher firing probabilities. And by using less electrical stimulation, there'll be less current spread. And by using less light, we're using lower power, which could mean that there's less power usage, it's a bit safer. So let's see what hybrid stimulation looks like. Using the patch clamp electrophysiology methods in the cultured spiral ganglion neurons. The hybrid stimulus consisted of a subthreshold optical pulse and a subthreshold pulse, and an action potential was recorded, while the individual pulses didn't evoke an action potential, being subthreshold. And so when we had a look at how we can combine the electrical stimulus and the optical stimulus, and these are showing the percentage of threshold that we can use, so they're all subthreshold here. The electrical stimulus could be as low as 20% of threshold, for example, if it's combined with a 90% optical threshold. So these are the range of stimuli that could be used to elicit action potentials in these cultured auditory neurons. Again, so maybe the optical stimulus is 60% of threshold, and it would be combined with, say, a 60% electrical stimulus. So now in this recording trace, you can see that having a look at these subthreshold depolarization events, you can see that they do look a little bit different. So we can zoom in on those. And this graph shows the membrane potential changes during subthreshold electrical and optogenation. And you can see that the ion channels have quite different opening and closing. This is here in the blue. You can see that it's extended in duration. And we can take advantage of this when we're using hybrid stimulation because we can actually delay the electrical stimulus relative to the optical stimulus. So say we had a 1 millisecond optical pulse, we can apply the electrical stimulation after that optical pulse and still get that increased firing probability. So this in this graph here, you've got the 1 millisecond optical pulse shaded in blue, and the firing probability was highest when the electrical pulse was applied at a delay after that, even up to 20 milliseconds for this cultured neurons. But what about in vivo? The timing and the kinetics are a little bit different, but the story is the same. So we have looking at multi-unit activity in the inferior colliculus. When we have a subthreshold optical or electrical stimulus, we don't get activity. When we apply the hybrid pulses together with no delay, so they're starting together, and there's no activity. It's really when that delays are applied such that the electrical stimulus is happening at the end of the pulse or after the optical pulse. This is when we're achieving this sparking activity with these subthreshold stimuli. Another way to look at it is the way the light influences the electrical threshold. And across the different delays. When there's no delay, the light is barely influencing the threshold of electrical stimulation. But when we are applying delays of up to 2 or 4 milliseconds, the relative electrical threshold is reduced to half. And this is because the light is essentially sort of priming the tissue to be more ready to fire. So the threshold for electrical activation is lower. So you'd expect this to have an influence on the spread of activation because we're not using so much electrical current. So having a look at this, we're now using a set delay of 1 millisecond. So that is the pulses are ending together. We used a fairly simple stimulation setup consisting of an optical fiber and a platinum wire through the round window of the cochlea. So it's not quite as optimized as when we're using the LEDs. In fact, if you look at the optical alone, it's not too dissimilar really to the electrical stimulus here. But for the hybrid stimulation, we were achieving a narrower spread of activation, and this is mainly due to that reduced electrical thresholds. So the results imply that we can use light to enable us to use less electrical current and reduce the spread of activation. So yes, hybrid stimulation is allowing us to use the electrical stimulus without significantly impacting the spread of activation. So we can use the electrical to drive the action potentials. So does this improve the temporal properties? So let's have a look at that. We've had a look at this in vitro and in vivo. So looking at the in vitro study, still with our rather sluggish opsin of H1R, using that rather sedate pace of 20 hertz stimulation, and that 10 pulse trains of suprathreshold optical stimulation, so the optical is at 110% of threshold. Optical on its own, as I showed you before, is only one action potential. But then as we add electrical stimulation at just, say, 20% of threshold, or 25% of threshold, or 30% of threshold, we're now seeing the spikes for every stimulus. And in the intact auditory system, we can see that the maximum following rate for hybrid stimulation is more than 3 times that of optical stimulation alone. And here it was this maximum following rate is defined as the rate for which 80% of the triggers had at least one spike in response. Now, this was measured at the level of the IC, which is 4 synapses away, and it's a rather indirect measure of the effects of optical stimulation on the auditory nerve. So again, looking at the data that Elise AJ has been doing, these are the compound action potentials recorded in response to optical and electrical stimulation, as well as hybrid, which I'll show you in a moment. And we're looking again at that ratio of the peak-to-peak response amplitude of the last response and the first response at 100 pulses per second. And as you can see, and as you recall, it diminishes during the burst. And as we use hybrid stimulation, when we've got when the optical part of the hybrid stimulation is low, so below threshold or at threshold, you can see that the responses are now looking more electrical-like. And as we dial up the light part of the hybrid stimulus, you can see that it does trend towards becoming more optical-like. But it's really these ones that interest me the most, the ones that are more electrical like. And as we know from our spread of activation study, when we're using threshold or below threshold light, this is when we can still influence the spread of activation. And also with the temporal spread, that is the difference in the timing of the response. You see with electrical only, it was pretty similar between the first and last, whereas for the optogenetic stimulation, it was poorer. But as we hybrid stimulation, again, we can sort of get that range of responses, and it depends on the optogenetic, or sort of the level of optogenetic or optical stimulation that we're applying within that hybrid pulse. So all in all, I think with the hybrid stimulation, what we're really trying to achieve is a little bit of the best of both worlds, where we're getting that good spatial resolution and good temporal resolution. And as you can see from those studies, it does depend on the level of optical stimulation that is applied, and we can certainly hopefully find that nice sweet spot. So just in the very final moments of the lecture today, just want to talk about the development of these translatable opsin expression systems. So we can't be using the transgenic mice. We need to use something like a adeno-associated virus to introduce the opsins. Spiral ganglion neurons in mice are not that easy to transduce. So there's been a lot of studies and so some work that we've done where you can, say, inject into the round window, or you can inject into the semicircular canals, or inject into the round window and generate that fluid flow by making a hole in the semicircular canal. So we did a bit of a study in mice to see how we could transduce the spiral ganglion neurons. And by far the most successful system is to inject into neonates to achieve a high percentage of spiral ganglion neurons which express the opsin. But in the adult systems, the one that achieved the best results, at least in our hands, was to inject through the round window with a hole in the semicircular canal. But it's not quite as successful as the neonate. So these are the results from the neonates, looking at the basal, middle, and apical turns. You can see there's a bit of a gradient of expression, which was highest in the apical turn. And we also did see some expression in the contralateral. The same thing was in the adults, where there was this gradient of highest in the apical turns and a little bit of expression in the mid to basal regions, with no transduction of the contralateral cochlear in the adult. And the level of expression, as you would expect, does influence the optical thresholds. So when you have no transduction, you have no activity. And when you've got low transduction, you've got very high thresholds. And as that level of transduction increases, so we can reduce the threshold. And I've compared it here to what we saw for the transgenic mouse, which is kind of like a 100% expression system. But it's not just mice, and certainly the research teams in Germany have been using gerbils, which have been injected with the AAVs as well, very successfully achieving opsin expression in the spiral ganglion neurons, and as I mentioned, getting that really nice optogenetic stimulation. How would it work in humans? I guess one study that I did find, which would be the the next best thing, which was to look at the non-human primate. An AAV was injected. This is not with an opsin, just with a reporter gene. They did find that while there was very high expression in the organ of Corti, there was also some expression, pretty nice expression in the spiral ganglion neurons here. And this is just with an injection through the round window membrane into the scala tympani, into the perilymph. So that does suggest that it will be possible to transduce the spiral ganglion neurons with a fairly simple injection that doesn't involve too many holes as I was creating in the mouse. And very, very briefly then, obviously that we also need to develop the technology of an optical cochlear implant. And again, there's some very nice work coming from Germany in this area, where by using the LEDs, there's certainly potential to have a lot more channels. And the things we sort of do need to consider, the optical cochlear implant is flexibility, the direction of the light emitters and how that's going to be sort of controlled, and perhaps externalizing the electronics to make it all very safe, which might require the need for waveguides and things like that. So there's a lot of considerations, but I just wanted to highlight some of those things. And of course, if we were to be considering hybrid stimulation, then we would need integration of LEDs with electronics. And what we're doing in the mouse for this is providing these alternating electrodes and LEDs. They're a little bit hard to see the LED through the silicone here, but there's 3 LEDs I think here between the 4 platinum pads. Looking at techniques about integrating those technologies and also the best configurations for those. So what I really wanted to highlight today was the work that we're doing with hybrid stimulation to help reduce the spread of activation compared to electrical stimulation alone, whilst increasing the reliability of firing compared to optical stimulation alone. And I absolutely acknowledge that we used a very slow opsin here in all of our studies, and that opsins such as Kronos can give very nice firing rates, which are getting towards what you would use in a cochlear implant. And we're really keen to see if we can use that technology with Kronos as well, and see, have a look at the sort of firing rates and reliability that we get with some faster opsins. And with hybrid stimulation, other things that we really like is that it uses very low power, because you only need to use levels that are near threshold, which can give safer and more efficient than optical stimulation alone. And that acknowledgement that to use this kind of technology, you would require to be expressing opsins in the system with AAVs, and that we need to be developing these flexible light emitters and integrating that with electrodes as we go. And that's how I'm going to leave it there. And I really thank you for listening, and I'd love to take questions.
Thank you, Dr. Richardson, for this wonderful talk. We have a question here. Well, actually, 2 questions. So the first is from Brian Finks. Thank you for your interesting talk and beautiful work. One basic question is how to do optogenetic and hybrid stimulation compared to tripolar stimulation. Also, if tripolar stimulation is not beneficial compared to monopolar, why are you optimistic that other mechanisms of a spatial restriction of excitation will be an advantage?
Yeah, thanks for your question, Brian. So how does it compare to tripolar stimulation? It's not something that we have directly compared. So far we have looked at optical stimulation alone, electrical stimulation alone. And for the spread of activation for hybrid stimulation, so far we've done that using the optical fiber and a platinum wire, and we're just doing some work right now where we're changing that to the LED system, the multi-channel LEDs. In fact, it's a multi-channel hybrid array. So that work is still in progress. So in that sense, we don't know yet how it's going to work with, or what the results are going to be for the channel interactions. But why am I optimistic that it would work better? Sort of revolves around the fact that we might be able to provide the simultaneous stimulation, and because to improve, the representation of complex sounds, we need to be able to provide the high spectral and the temporal resolution. And it would be really nice if we can be able to use a multi-channel and simultaneous stimulation. And maybe correct me if I'm wrong, but I think if you're doing some complex tripolar stimulation strategies, you're using 3 electrodes, and maybe reducing that ability to provide that multi-channel stimulation. So anyway, yeah, we're really keen to see if we can increase the number of independent stimulating channels using light, because it is so precise in its area of activation.
Okay, thank you for your answer. So we have another question here from Elizabeth Kert. In the experiments where you co-activate neighboring LEDs in the multi-LED array, did you always use simultaneous 1 ms stimuli at different intensities, or did you also vary the stimulation pattern for the neighbors?
I believe that we were using 1 millisecond stimuli on both of the LEDs. So what happened there was, one of the LEDs had the 1 millisecond pulses at a set intensity, and the second LED had the 1 millisecond pulses at a range of intensities.
Perfect. Dr. Bob Carolon raised his hand.
Yeah, can you hear me?
Yes.
Oh, great. Thanks. It's really exciting work. I just want to make a couple of comments. So one of them, I think, following Brian Fink's point, you're using mostly when you evaluate spread of excitation, you're using the same methods which are used to evaluate spread of excitation for tripolar and focus stimulation. And we know that that doesn't really apply to humans. So maybe it would be time to start looking at either behavioral or cortical measures of spatial resolution or spread, because then you'll kind of really know. At present, all you know is that this method, which tells you that other things should work, shows you all things should work. So yeah, so that's
it'd be really neat, wouldn't it? Yeah, to have the studies.
Train your mice. So we have some. Because offline, we've got some ideas for kind of more objective, non-behavioral measures of spread, which you might be able to use. But my other point, which also relates to spread with the hybrid, which is I can see how you've nicely managed to reduce the spread of excitation by sensitizing a small patch of neurons near the electrode. And that would work for a single channel system. But imagine you've got a multi-channel system. So you've now got another electrode nearby, and you've also sensitized those neurons. So then the reduced current spread from the first set of neurons might well then activate them, because they're pre-sensitized. And the only way you could get around that would be to time the sensitization with the light, so that it was selective for each pulse. But then, of course, you're stuck with the temporal limitations of the light effect.
Yeah, look, there's no doubt that we're using sort of fairly basic setups as well, and the way we're setting up the experiments, which is to enable us to sort of develop all the basics and or find out the sort of whether the whole concept is working, but actually implementing it in a sort of multi-channel complex system will definitely be a challenge. But one of the things that we're quite interested in is sort of, I don't know why I thought of this right now, but for example, you can apply things like current steering in addition to like over the light source. So you know how we had a alternating LED, platinum electrode, LED, platinum electrode. And, one could argue that the actual hybrid channel is not directly sort of on top of each other. They are side by side. So it might be really neat to do current steering across a single light channel and see the influence on the thresholds in the middle, and see if you can sort of sort of steer the current to be over the light, things like that.
Okay, thank you. We have one more question from Keith Abbey. Great talk. Much thanks for your time. Is the amount of electrical versus optical stimulation dependent or at least correlated with the activation rate of a chosen opsin?
Yes, I would say that it is. It's very much dependent on the opsin that we use. Opsins have different sensitivities to light. Some of the faster ones do have lower sensitivities to light, and you have to use more light to activate. I don't know about the electrical being dependent on the activation rate of the opsin. But yeah, certainly the choice of opsin is going to be very influential. And so I was just laughing at Bob's comment there. So the choice of opsin is going to be quite influential on the amount of light that you use, obviously the color of the light and everything like that.
Okay, perfect. So we don't have any other questions. So thank you, Dr. Richardson, for your time. We know that it's pretty early in Australia, so thank you so much for coming. Thanks to all of you for your participation. We'll have again this international series next semester, maybe in August or September. So all of you are pretty welcome, and have a wonderful evening or wonderful afternoon here in the other side of the world. So thank you, and thank you, Dr. Richardson.
Thanks for having me.
Bye.